excellent speakers. Our next speaker is Steve Chang. He did his undergraduate at Washington University. He did a postdoc at Duke, and then he moved to Yale, very, very sterling credentials. And he tells me that he has actually been working in neurobiology of monkeys through all these different places and these different positions. And I must say, I looked up his webpage and I keep seeing young researcher of the year, promising young researcher, and many things like that. So I think we have a lot of very good work to look forward to hearing about. Steve?
Hi, everyone. So I think this is a last talk of this summer school. So I'll try to make it kind of fun for you all. So neurobiology monkeys thinking about other monkeys. Obviously, we cannot ask them what they're thinking. So the best way we can do is try to probe their behavior that passes several tests that have to be true when they're considering others. That's what we're going to talk about today. So these are just the videos of wild rhesus macaques in Cayo, Santiago. You can kind of see that one group is defending another group that's invading their territory. But you can kind of see they're checking back and see who's got my back. And they kind of, threaten together. They're definitely aware of self and other. And this is kind of actually a good example of cooperative behavior in rhesus macaques. Also, if you walk around this island, you see a lot of times monkeys spend really a lot of time grooming each other. This is actually occupies big chunk of their daily routine. Especially this one feels like he's having a really good time. Yeah. Okay, so rhesus macaques are really have a, to me, very interesting social behavior, but not interesting because they're really weird and bizarre in a weird way, like a New World monkey sometimes, or eusocial insects. They're very different, right? But rhesus macaques' social behavior is kind of very similar to us in a lot of rudimentary ways, but maybe they're a little bit more of a despotic, aggressive version of our social behavior. They live in large hierarchical organized groups, about 50 to 70 members in a troop. And interestingly, males leave their troops upon puberty. So, males have to explore, go out, find a new troop. And when you're encountering new troop, that's a big challenge because they're very despotic species. So you have to really recognize who's the dominant one, who's your foe and friend, basically, to integrate within a new society. And as I'll show you, they show many of the basic forms of high-level social cognition in humans. So rhesus macaques are really an excellent model of the human brain. In particular, this is a rhesus brain, and this is a human brain. They're actually a lot smaller. They're not the same size, but this looks like the same size. But they have very homologous areas, like a Broadband structures map nicely onto monkey brain. And really highly functional similar brain regions exist in monkeys and humans, especially the prefrontal cortex. So, if you're studying prefrontal cortex, once you go to the rodents level, there are very different types of prefrontal cortex. They're agranular. But in the rhesus monkey, you have a granular and agranular prefrontal cortex, just like humans. And hallmark of human is we're really visually driven species. So are the monkeys. This is all the colored area. Well, this whole thing is the brain, basically, stretched out and flattened out like a pancake. And what you see is that all these colored area are majority visual areas. So that occupies almost half of the whole brain. That's a big part of the dedication to the visual system. So how to study social cognition in monkeys? You can use a majority of the same techniques that you use in human subjects. Here's an example, kind of classic example now, patient SM. She had a bilateral lesion to amygdala, and her lesion, she got famous because her lesions are so focal, very tightly to amygdala and not hippocampus, which is touching the amygdala. So it's very specific. And this group from Ralph Adolph's lab and Antonio Di Masio's lab in early 2000, they looked at how people look at these different emotions, and they tracked eye movements of the control. In this case, the control brain damaged patients, but not in the amygdala. And they look at a lot of times they look at the eyes. Patient SM, if you look at carefully, a lot of dots are missing. A lot of fixations are missing from the eye region. So you can kind of study this kind of thing in rhesus monkey as well, because this is one sample size of one. But in rhesus monkeys, you can actually cause exocytic lesions so that you can selectively lesion the amygdala in both directions. And different color just means number of subjects. So red means all four monkeys had the exact same lesion. Green means this tiny bit, only one monkey had the lesion there, for example. So you can look at the eye movement scanning pattern of these images. They used monkey faces at this time. What you can see is that in the lesion, in the control animals, you can see monkeys look at the eye region a lot, but not much at the mouth. But in the lesion animal, you have much more shifted attention to the mouth in this kind of paradigm. So you can kind of use a very similar approach to learn about human brain. And for example, if you want to, if amygdala is important for paying attention to the eyes, what's the mechanism? Then you can now record for neural activity to see what kind of computation is going on there. Here's an example. This is actual real commercial I encountered. I later found out this was aired during one of the Super Bowl event, which the ad space is really, really expensive. So they put best things out there. But anyway, I thought this was very funny. So these are actually scanning patterns.
I hate Mondays.
Yeah, I'm the worst.
No worries, man. Everything will be all right. Yeah, man. Oh, don't fret me, brother. Sticky man come soon. Yeah. Wicked coffee, Mr. Jim. Julia, turn the frown the other way around.
These are two different subjects scanning patterns.
So, right? Yes, I. The land of 10,000 legs. The gopher state.
So in conclusion, things are pretty dismal.
You know what this room needs? A smile. Who wanna come with that? Traveling along with a song that we're singing. Come on, get happy.
You guys are 3 minutes late.
Don't be no cloud on a sunny day.
Yeah, chill with stuff.
Sir?
Respect, bossman.
It's a Volkswagen commercial.
German engineer.
Anyway, so the reason I put it up there is funny, but also, you can kind of, you'll look at very similar places, and the social attention is just a term they use to how you're paying attention to different parts of other agents, actually gives a lot of information. It's almost like a prerequisite before you even begin to understand others' mind, because you have to first engage in the right place. These are the monkeys now watching movies. This is work by Freiwald's group. And these here is just showing the regular videos they watch. And this is the eye tracking of these while the monkeys are watching. And you can see that, where they're looking, direction of the gaze. Right? So it's not like, they're really paying attention to places that are, ethologically meaningful to them. And now you can, study this kind of thing. This group particularly used the monkey functional neuroimaging to find out, are there specific brain regions for looking at social interaction versus some non-social object interaction, for example. So we tried this kind of experiment. But we are very interested in spontaneous gaze behavior. It's very different from like you train monkeys to do something or look at something. But instead, we didn't tell them anything. We didn't train them for anything. But we just wanted to see how the two monkeys are interacting dynamically and contingently. So, we have to first calibrate these guys. So you have two animals in there, you calibrate, and both of them are eye tracked at 1,000 hertz, so we can have a very fine resolution at 1 millisecond. And then after calibration, we just leave them there, and they can fall asleep if they want to. They can look around, they can look at each other. As a control, we were very interested in comparing this picture condition, where the majority, almost every study until this moment has been done looking at pictures of monkey faces on the screen. So this is more like a static only condition. There's no dynamic component there. There's no contingent component there, because what he does doesn't change anything of this guy. Movie condition we added there, and this is actually movie footage of this other guy in the screen. The interesting thing about this is dynamic, but there's no contingency. He's not gonna contingently change his gaze or something like that because of him, whereas this condition has both contingency and dynamic components. So we're interested in how social attention is deployed in these kind of three comparisons. This is a single session example from the live, and these are where the monkeys are paying attention. The red means higher fixation in that case. And you can kind of see generally they look at the eye region more of this and look at the other parts too. If you quantify this in a time averaged fashion, so we're not looking at dynamics yet, we're collapsing across time domain, and we see that all picture condition, movie condition, live condition, generally they all look at the eyes quite a lot. But there are some subtle differences that are highly significantly different. For example, if you look at the proportion of fixation to the eyes, live condition has the highest proportion of fixation to the eyes compared to movie and compared to picture. And also the gaze spread, this is just how focal it is to the eye region. Live condition is the most focal, whereas things are more dispersed in the picture condition. Duration of fixation was also different. Live condition had the shortest duration fixation per fixation. This was not due to some general non-specific arousal, because one possibility is if there's another monkey there, you're more aroused, so you might look at things differently. But that prediction would predict that they'll look at the most everything. But the raw number of fixation to the face was actually highest in the movie condition, and the total raw number of fixation was actually not different between movie and live. So using this kind of task, now we can actually test social relationship is reflected in this gaze behavior. So in lab setting, you can kind of, familiarity is already there. We can call familiarity as cage mates versus monkeys that are in a different unit. They're all in the same colony. They know each other, all of them, but it's a matter of how familiar you are with another monkey, because they can interact all day long, but they can't. Dominance, on the other hand, you have to quantify dominance because in male monkeys especially, male macaques, their dominance hierarchy is kind of malleable. They have a matriarchal society, so female macaques born into higher rank, and they stay there very steadily. Whereas the males, there are a lot of fights, and the fight often changes structure. So to really know males' hierarchy structure, you have to test them in the lab almost every 3 months. Otherwise, it might have shifted around. So we do two different ways. We do food grab test. We basically give a piece of grape they love, and then the dominant one will grab it, subordinate one will not. And also there's a controlled confrontation task. You just put them together for like 5 minutes and score how many times one monkey is directed towards and how many times another monkey is directed away. And dominant ones are the ones directed towards more. Because directing, looking directly and directing is in monkeys is a threatening gesture. It's a sign of a challenge. So subordinates don't do that much. If they do, then in the wild they get beat up and get kicked out and such. So you can use this kind of two different tasks to quantify dominance. So to look at the non-time average way of looking at things, to look at dynamics, you can kind of score over time, when each monkey is looking at the other monkey's, let's say, eyes or mouths or ears, whatever you want. And each dot is when, in this case, this monkey is looking at this monkey's eyes. And this is when this monkey is looking at that monkey's eyes. And you can kind of draw a tick mark there. You can make this binary, just like a firing rate of a cell, so ones and zeros. And now you can align the two, and whenever the two are aligned perfectly in time, that's a mutual eye contact. And this little bulging out square is the duration of the mutual eye contact. When someone else breaks, now that mutual eye contact ends. So you can take this kind of approach to make peristimulus time histogram, just like when you do it with a neural spiking activity. So here's the example from the day 2 days. I'm now aligning the gaze proportion of fixation to the eyes of another monkey as aligned to the mutual eye contact event. So in the beginning, of course, it's really high because that's how it's aligned. But over time, you have this nice decay activity that fit really well with exponential decay. And I'm coloring two different tones here, dominant one and the subordinate one. And you see that over here, dominant one has a higher proportion of returning back. This is just 1 day. This is another day, and you see a very similar pattern aligned to the mutual eye contact. So these are just 2 days data, but we collect many, many days across many, many pairs. So we want to quantify that. So this is from 10 unique pairs of monkeys, which gives you 20 unique behavioral perspectives. Again, it's aligned to mutual eye contact. And now you can see the dominant, the subordinate one is the orange, dominant one is green. You see that after mutual eye contact, the proportion of looking back at the eyes is much more elevated compared to subordinate animal. Now we can start to quantify this more carefully. So when we now look at this, this was aligned to mutual eye contact. When you align the same data, but now aligned to non-mutual eye contact, this is when one monkey is looking at the other monkey, but the other monkey is not looking back. So we have, from the eye-tracked monkey's point of view, you're looking at the other monkey's eyes in the both settings. In one setting, the other wasn't looking back. The other setting, they are not. And you see that this kind of dominance-induced change in this apparently gain kind of thing is not there at all. And you don't see that in the picture condition or the movie condition, even if you align to the mutual eye contact. So this dynamic seems to at least be, by design here specific to the live condition. And we think it's probably because for this kind of dynamics, this kind of effect to be seen, you want to have both dynamic and the contingent conditions. These are just control that we can show that how specific it has to be to the eyes. When you increase the allowed window, the effect disappears. This is a population fit of the data. You can see that dominant one has a higher gain, whereas the rate is actually same. We quantify this. Rate is conserved, but the dominant subordinate differences came in the gain of the decay function. What about familiarity? Well, we see very different thing in the familiarity. We don't see the gain effect at all. But if you're more familiar, cage mates, you have a faster decay. It's more like when you really know someone, some other monkey really well, you have a faster, you lose interest faster, or your attention back to the other monkey decays faster than the monkey you're less familiar with in purple. Again, you don't see any of those effects when you align to non-mutual eye contact. You don't see it in the picture or the movie condition. And this was, again, specific to the tightness of the eye region of interest. And the quantification of that using the exponential fit is that if you're less familiar, more familiar, the gain didn't have any significant change, but the rate of the decay was faster if you're familiar. To me, that kind of makes sense. You already know each other much better. So up to here, I just want to set the stage for social attention being one of the really important thing before you get to thinking about others, which we can study using decision making, because we cannot really ask monkeys, but we can ask monkey to make a decision that impacts reward outcome, for example, for you and for the other. And based on what they do, we can kind of understand, oh, they have other regarding preference, or they have antisocial preference. And then we can now look at what kind of brain activities coupled to that. There are a lot of studies has been done in the non-neuroscience domain, more in the primatology tradition. I know Sarah was here, Sarah Brosnan was here like a few weeks back, last week or something, but she did a lot of work like this. So capuchin monkeys, they deliver same amount of food to another than themselves. They show stronger prosocial tendencies with others closer, like a kin compared to others. They also show inequity aversion, and they more likely to give high rewards when partner is present. All these are kind of bits of information that tells you that capuchin monkeys have other regarding preference. Other regarding doesn't have to be all positive; it can be negative too. So they care about the other monkey basically. Chimpanzee, this is not a monkey, this is ape, but they also have people studied a lot in the field. They prefer to deliver same amount of food another than just to themselves. They transfer tool use to the other guy, open door for the recipient when they need to get food. And this was an interesting one. Dominant monkeys are actually more prosocial than subordinate ones. And this is often called a directed downward hierarchy kind of effect. We actually see this in monkeys too. I'll come back to it. When the dominant one has nothing to lose, then they can actually give this prosocial gestures downward hierarchy. And there's also competition effects as well. Okay, so how to investigate social decision making in non-human primates from neuroscience perspective. One of these earlier studies, what we did was we asked monkeys to basically play a monkey version of a dictator game. Dictator game is from a game theory. One human just makes reward allocation decision to others. Others can accept it if they want to, but it's different from ultimatum game. It's just like very dictator-like, one-way transfer. So we have an actor. He's the dictator basically. There's a recipient monkey in the room, and they are about 100 centimeters apart. There's a dim light there so they can see each other. We don't want them to not see each other. And actor is the monkey that makes all the decisions, and they have a stimuli. Depending on the condition, he gets some juice. Depending on some other condition, recipient may get juice. Importantly, we have procedures here that we first train the monkeys what different colors or shapes. We also try with fractals might map onto the reward outcome concerning self or other. So this is basically just a simple class conditioning procedure. And what we do is the actor monkey, there's two types of conditioning. Type 1 is basically the actor looks at the fixation. There are eye movement-based tasks. And one of the cue comes up, and the cue might mean that, okay, reward will go to self. And the cue goes off, and then there's some time to look at each other, meaning that they don't have to fixate on the screen. There's a reward delivered, and this reward is 100% contingent on what you see over here. So it's a cue-reward association. And we do this for reward to self, reward to both monkeys at the same time, just to other or just to neither. And this is basically operant version of this. We have fixation, cue comes up, cue goes off, and there's some neutral target comes up, and monkeys just have to make an eye movement to realize that reward. So we do this conditioning for like a week or so, but we have to know that they understand. And they quickly understand self-both cues from other neither cues, because self-both cues, they get juice. It takes them 3 trials to know. But other from these two to these two, they immediately like these two colors, but not these colors. But the important thing is we want them to show differences in other and neither. In these both contexts, our self monkey is not getting the juice, but one time juice is going to the other monkey. So what we do is we look at the error rate. Error is just defined as breaking fixation from the cue. So when they see a cue that they don't like, they oftentimes just look up and break the fixation, and that's an error trial. So you can see that in the beginning, this is like first week of data. Monkey make a lot less errors in the self both compared to other neither, because they get juice, they don't get juice. But towards the end of the week or so, you start to see that monkeys are making less error when the reward is going to the other monkey than the neither monkey. So they're not really making a decision per se in the direct decision to give or not give, but they generally prefers, when they're not getting the juice, they generally prefers to give to the other monkey than neither. So once we see this kind of preference, we go to the choice task. So choice task is what you need if you want to directly probe their preference. right? So we have same condition here, but we also have a control condition where recipient monkey is replaced with an empty primate chair where monkey would be sitting in otherwise. And we have a little bottle here so that monkey can still deliver this juice to the empty bottle. The reason we did that is, if they actually prefer giving juice to the other monkey, that could be because the monkey is making something happen in the world, like delivering juice, versus neither is nothing happening. So we want to control for that kind of salience difference. So we put juice bottle here. So if delivering juice to empty bottle versus neither is more salient, they should still do that if the salience is explaining this effect. So the preference test goes like this. They fix it in the center, and two targets come up. Two targets can come up in any places in the circle. And importantly, they are reward matched. What that means is that they're always choosing between giving juice to self or both, and between giving juice to the other or neither. So self and both, actor monkey is always rewarded. So there's no harm or benefit here if you're just thinking about yourself. Other and neither, actor is never getting rewarded. So there's no harm or benefit of neither if you're just not thinking from your own perspective. The important thing is that if actor monkey is completely careless about the social situation, doesn't care anything, because of the task, the optimal behavior to maximize your own juice outcome in a given time is choosing randomly, because targets are always changing, because there's no differential in reward. So after they see the choice, they make an eye movement to choose one. Here they chose, I don't know, give to the other monkey. And the reward is delivered based on what he chose. So now you can look at the preference of this activity, this behavior. If you look at the other-neither context, monkeys are actually, actor monkeys definitely prefer to give to the other monkey than wasting it. Interestingly, in the self-both condition, monkeys actually are antisocial by definition of this framework. Like there's nothing to lose, but they're not going to share. So this is very interesting contrast that we've seen again and again in so many monkeys. But importantly, when the monkey is replaced with a chair and juice, we see basically no preference emerging from either context. Now you can quantify this making an index. It's just an index such that reward giving goes here, reward withholding is this way. In individual, these dots are different pairs of monkeys we tested. You can see that there's a monkey-dependent spread there, but they never cross the direction. And later we found that some of these effects are explained by dominance effect, and that's why I mentioned about the chimpanzee downward-directed hierarchy. Some of this effect here, monkeys actually, dominant actors are a little bit less, significantly less social when there's nothing to lose in this context. And over here, the spread is explained somewhat by, if you're more familiar, you are more donating to the other monkey more. Because we're using eye movement tests, we can also look at the reaction time. In psychology, reaction time has been a really good measure of subjective preference overall. So we can look at, if they choose self over both, they should be faster in reaction time. If they choose other over neither, they might be faster in other's reaction time. And that's what we see. This is the reaction time curve. And again, when the chair juice condition, we don't see this kind of monotonic increase there. We talked about there's some free viewing period where the monkey can look at each other. We can also look at, do they look at the other monkey at different times depending on what they choose? It turns out if they choose self and both, they look at the other monkey about 5% of the time. When they choose other, they look at the other monkey more than neither. And I think this is more convincing way. We actually also did a test so that one recipient is over here and the juice bottle directly is over here, so that juice is going into one of these places depending on what he chooses. Again, the non-social condition, you don't see this kind of this pattern here. But here, what you see is that if you look at looking at recipient condition data, then you see that monkeys look at the recipient most, and they peaks at other after choosing other. But if you look at the eye position towards the bottle, now you see that that increases the most at the niter condition. So this tells you monkey exactly knows, well, exactly to the degree of 40% of the time how they express this, knows where the juice is going. So they're definitely understanding the task. I know Larry Young was here a little bit, but I want to also talk a little bit about oxytocin, because oxytocin worked pretty interestingly in our task as well. So oxytocin is a Greek for quick burst. It's actually the first hormone to be synthesized ever. This one, and this person won Nobel Prize for it. It's synthesized by a single gene oxytocin gene in the hypothalamus, supraoptic, and paraventricular nuclei. And it's stored in posterior pituitary and released in dense core vesicles. It's really well known for lactation and birth. This has been known for a really long time. Lactation and birth, uterine contraction, sexual behavior. If you're going into labor, but things are not moving forward, and you have to move it forward, then they give you pitocin. Pitocin is basically oxytocin you get in the hospital. Then you start labor right away. Those are peripheral actions that have been known for a long time. Central nervous system, however, has been up to about 5-10 years ago, it's been very mysterious, but now we are understanding much better. Thanks to Prairie work, it really changed how we think about oxytocin. And also, across humans and monkeys, we learn a lot about oxytocin role and prosocial tendencies. And there's also some interesting promise, but also not that good of a promise about oxytocin in treating social disorders. Some successful, some's not. And more recently, this is a more recent trend. The people who work in the optogenetic circuit world are very starting to get interested in oxytocin, and we're learning a lot about the very specific circuit mechanism of oxytocin these days. So what happens in this test if you give oxytocin to these monkeys, this actor monkey? So what we did is we used, instead of intranasal oxytocin, which is a spray, monkeys, that doesn't work that well, because monkeys don't do this for you, so it just leaks right out. So you have to use pediatric nebulizer to be really effective, and we can just inhale this. And this is just a picture of a little kid taking it. If you have allergy, if you go to the emergency room, they give you like Arbitral, which is nebulized. So you can nebulize oxytocin this way. We wait 30 minutes, because according to guinea pig research, it takes 30 minutes to go to the central nervous system. And we actually, and then we did a task, right? To see how this changes the behavior. So up to, actually, like interestingly, surprisingly too, up to this study came out, no one has actually documented intranasal oxytocin or any nasal route oxytocin gets to the brain. So we actually said, okay, we got to measure this. So we did cerebrospinal fluid measure of oxytocin before and after saline and oxytocin condition. So you see that this is the baseline CSF oxytocin level for the saline treatment. And when you give the oxytocin, it increases about 2-fold, 2.5-fold. And this is just the typical dose people use, 24 IU. And just to confirm that it get there. And now we can look at the preference index over trial number. The reason we're looking at the trial number is that there's a time course effect of this hormone. So here, this is choosing other versus neither. The fact that it's positive means they prefer other over neither. The gray is the saline condition, red is the oxytocin condition. You see that in the beginning, they're very similar. But towards the end, the oxytocin is becoming, resulting in higher other regarding prosocial choices here. Interestingly, if you look at the other condition, in this case, we compare self versus other and self versus neither. This is interesting because monkeys, here, self versus other, self versus neither, the other option has something to lose, right? The actor monkey never gets the juice if they choose the other option. Well, they never choose it, really. They're almost 99 or 98% of the time choosing self over other, self over neither. But if you actually break it, look at this more zoomed in, you see that some differences emerge. So this dark green is the saline condition. But even just the noise embedded in this preference gets flattened out with oxytocin. So what this tells you is that, it's not that oxytocin making you prosocial all over the place, but really is context dependent. So in my theory always has been that oxytocin doesn't create anything. Oxytocin is a modulator that amplifies pre-existing preference. So if you have a prosocial preference in one context, oxytocin might help you there. But if you have kind of nasty attitude on the other, it might make you even nastier. And that has kind of context-dependent effect. It makes a lot of sense to me because it's a hormone. It's a neuromodulator. It turns up gain up and down rather than creating new computation or something like that. And this kind of context dependency has been seen in humans and as well as in rodents as well. So we have a choice trial, and we can look at the social gaze following that on choice trial again. Oxytocin actually increased looking at the other monkey, up to about 44% of the time. And we can actually look at this over time again. And again, in the beginning, you have a very similar with salient and oxytocin, but kind of halfway through, looking behavior kind of increases. I don't know how actually this is directly related, but the timing of increase due to oxytocin, looking behavior, and the choice preference kind of happens around the same time, kind of midway through the task. Interestingly, we also had Q trials. So exactly same reward outcome is delivered to the other monkey. It can be self, both, other, neither, whatever. But monkeys are not making a choice. So this is we making the choice for them. When we do that, we don't see difference in social gaze to the other monkey, which suggests that this increased looking to the other monkey was specific to after you made that happen, after the actor monkey made that happen. So it's kind of linking interesting perspective on, providing interesting perspective on this. It's not just that oxytocin is making you want to look at the other monkey drink more because maybe that's more rewarding. It's not that. It's almost like you want to look at the other monkey drink more after giving the juice to them. So I want to do a quick detour, a very quick detour of the interaction between oxytocin and opioid systems, because this is kind of really understudied system, and we find some interesting effects here. So this is a paper by Bickner and Lang, and I think this paper is really cool. What they did was, they had a posterior pituitary on a dish, and you stimulate it with electrical stimulation, and you can cause oxytocin release and vasopressin release, which are tightly related. But interestingly, so this is the, you stimulate, you get an increase of release. But interestingly, when you bathe the tissue in naloxone, which is mu-opioid receptor antagonist, you have, and apply the same microstimulation, you get like crazy high release of oxytocin without changing AVP, vasopressin release, which is kind of same level as the baseline. So this is specific to oxytocin release. Something about blocking mu-opioid receptor is like making oxytocin release go really high. So we thought about interesting experiments based. This came out in 1982, but we just did this study like 2 years ago. But the interesting idea was, one of the issue with oxytocin is that intrinsic oxytocin is sometimes works, sometimes doesn't work. It doesn't really effectively, robustly change your behavior. And we thought maybe we should take advantage of this already existing physiological relationship by delivering oxytocin in combination with naloxone, such that in your body, let's really turn up the oxytocin release and see what happens. Just to support this idea, chronic morphine reduces oxytocin release in humans and animals. Morphine delays parturition during birth, inhibits lactation, and it is due to reduced oxytocin release. This can be all reversed by oxytocin. So we tested oxytocin and naloxone's effect to see whether this is the hypothesis we're testing. If this attacking the system, coming at the system in two different angles can really help, then giving oxytocin alone or naloxone alone, if you sum these two, the combination should have a bigger effect, right? If this is linear, meaning oxytocin plus naloxone is same as oxytocin and naloxone together, then it's not that interesting. Then you're just upping the dose. But if this actually is greater than the sum of each effect, then now you have superlinearity, which gives you that you're tagging into something that you cannot tag by doing one or the other. So in the live gaze interaction context, as shown below, a bit before, we do the same thing, but we did saline, naloxone, oxytocin, oxytocin, naloxone together. And to me, this is an interesting plot. If you just look at the oxytocin, naloxone minus saline, you see that they, compared to saline, they look a lot more at eyes. They look actually less at mouth, which is interesting because oxytocin, naloxone together is shifting your attention more to the eyes. But this is the main quantification of the main finding. If you actually compare oxytocin, naloxone to the sum of each drug given separately, you still see this redness in the eye and the kind of bluiness in the mouth region, suggesting that this, compared to these two sum, is still surviving a further increase in the attention to the eyes. You can do this at the non-time averaged way again, dynamics. So now we cannot align to mutual eye contact. And what you see is that after saline, you have this nice decay. Same thing for naloxone, same thing for oxytocin. There's a slight change here. But if you give oxytocin naloxone, you have a lot more increased proportion of looking back at the other monkey. And we can quantify this at a certain interval. Then this is the unity line. So everything above here is a supralinearity. And in fact, it is. Interestingly, now when you, again, align this to non-mutual eye contact, then all this, the benefit of combined drug is not even there. So it's something about mutual eye contact that makes the oxytocin-naloxone combination to come out. We also did this in a different context where we just deliver mutual reward to the two monkeys. So no behavior at all, just all of a sudden there's a cue and here you go, get two juice, one juice for you, one juice for you. And this time you can look at the proportion of looking at the mouse. The oxytocin naloxone increases greater than saline or oxytocin naloxone alone to the eyes. Again, when there's no reward, this is no difference there. And point out mouse here because before it was attention to the eyes, but in this context, when the juice is going to each other monkey, now where the action is really is the mouth. So when you do the same thing, but now looking at the eye, you don't see any effect of this mutual reward. So, so one way to say it is they know which part of the face is important, and that's where the oxytocin naloxone effect is boosting the effect. So now going into the social decision-making part. This is the last section of the talk. So in humans, a lot of people have studied human fMRI of social decision-making. And these areas, anterior cingulate cortex, anterior cingulate gyrus, ventral striatum, amygdala, orbital frontal cortex, all these areas are again and again come up. It's very reliable, actually. And what's interesting is that these areas are known to be reinforcement-related areas. If you do any task with reinforcement-driven decision-making, that's nothing to do with social behavior, you get very similar activity. So this kind of links the interesting idea that a lot of social behaviors are reinforcement-driven. So we wanted to test this at the single neuron level. So the hypothesis we had, when you stick an electrode in these brain regions and get signal, we wanted to see some kind of correlates of social cognition in this case. And the hypothesis outcome in the reward outcome encoding as a test case is the following. We can have reward delivered to self, another monkey, or neither. And let's say I found a cell, I'm recording it, I'm trying to quantify the activity difference of these three different conditions. And the hypothesis is that you might find this kind of neuron. There's self-reference. This is basically not socially relevant neuron. When self is getting the juice, it gets juice response there, but nothing for the other conditions. Again, this is another type of self-reference type of neuron. This time, as long as you don't get the juice, it has elevated activity, but when you get the juice, it's lower. So this is more like the reward omission signal, or we call it sometimes foregone signal, very much related to prediction error as well. But most interestingly, we're looking for this type of neuron, other reference. The neuron that's exclusively encode the reward outcome of the other monkey, but not coding self or neither. And this, we might also find this kind of neuron, both reference. This neuron might be mirroring your reward and other reward in the same way. so that if I'm, watching this neuron, I actually might have no idea which monkey got the juice because they both have the same response. But I know at least one of these guys got the juice because when no one gets the juice, it's quiet. So those are the tests, what we tested. So this is a summary of a single neuron examples recorded from anterior cingulate cortex sulcus area, ACCS, orbital frontal cortex, OFC, and anterior cingulate gyrus, AC gyrus. AC gyrus is a part of the human medial prefrontal cortex. So this is where a lot of interesting things are found in humans. When we do that, this is representative cell in OFC. When actor monkeys in the juice, you have a strong reward response. This is aligned to the time of reward onset after the actor decides to give himself the juice. But when the other monkey gets it or no one gets it, the activity is not sustained there. So it was coding more like the self-reference received type neuron. In the ACC sulcus, this is also sometimes people call it dorsal ACC, DACC. You see that reward omission kind of signature. As long as you get the juice, these two different red lines, the activity doesn't get elevated much. But when other monkey gets it or no one gets it, it has a higher signal there. But here we don't know whether other monkey got it or no one got it because the two are not significantly different. Most interesting cells are found in AC gyrus in our hand, and other people have replicated even in human recording studies now. Well, in AC gyrus, you have neurons like a self-reference neuron like that in OFC that signals for self, but you have this type of neuron that signal exclusively when the other monkey gets the juice, but not for any other conditions. We also see both reference neurons that are signaling as long as one of the monkey gets the juice, but nothing when no one gets the juice. Recently, some other group did this kind of experiment in human neurosurgery patient, and also found that this kind of region encodes other persons' real outcomes. So if you quantify all the recorded neurons across OFC, AC sulcus, AC gyrus, you see that red is high in the OFC and AC sulcus. These are mainly received type neurons. These are mainly foregone type neurons, whereas the AC gyrus used to have them, but it's reduced much. In comparison, if you look at other reference and both reference neurons, we can call this something to do with other monkey. Then OFC and AC sulcus have a smaller proportion compared to much higher proportion in the AC gyrus. And this classification is done, with a super conservative way to test it. For example, if you want to be other reference, it has to be not significant for self, not significant for another, and only significant for other, for example. So AC gyrus, it's not the most popular brain region, but in fact, it kind of is because in rodent work, people often look at infralimbic cortex, prelimbic cortex. In rodents, they don't have sulcus and gyrus division. Rodents is basically all gyrus. So a lot of studies have been done, but in primate species, including humans, much less work has been done. But this is macaque monkey, this is human. You see very similar subdivisions in the ACC medial prefrontal areas. And this is just to demonstrate that, one color is human, another color is monkey, putting a little seed in AC gyrus and how it connects to all the other brain areas. This kind of spread is very similar, literally overlapping, suggesting that the connectivity, this kind of functional connectivity patterns are very similar between monkeys and humans. And again, also the AC gyrus happen to be connected with other important reorder-related regions, as well as like a TPJ, where people have been finding theory of mind kind of activity in humans. Human fMRI studies have found AC gyrus to be important for representing others' reward, others' net value in some kind of game, others' prediction error, what other person was predicting but didn't get, and also others' pain. So there are a lot of interesting effects there. In a competitive strategic learning situation, influence of others and the parameter estimate from this AC gyrus areas are correlated. And also when you take advice from others to make a decision, there's a correlated activity of how much you're going to take advice in the gyrus, in the red here, whereas the green is the sulcus, which actually reflects instead your own reward history. So there are some interesting divisions there. So I want to also talk about the social error monitoring, because like if you're understanding other monkeys' mind and playing this kind of game, it's also important to think about what other monkeys are doing really well, right? So this is studied by Masaki Isoda's group, and it's a very clever task. You know, two monkeys are, the goal is to understand, do certain neurons represent others' action differently from my action? And also, do neurons care or represent when other monkey makes a mistake compared to when I make a mistake? So that is this turn-taking game. Two monkeys are sitting at each other. They press a button to start. You know, each monkey takes two choices, green, red, and one of them is rewarding, one of them is not. And after making a choice, roles reverse. But the critical thing is, when roles reverse, you know, this monkey now has to know what this monkey chose last to make a right choice. Otherwise, you know, he has 50% chance of getting it right. So this task forces this monkey or this monkey to pay attention to the other monkey's action and monitor what happens. For example, here, if the yellow was the right answer, and that was the last time he chose, the next time he will go for yellow because that was a rewarding option. But let's say yellow was not the right option here, and then rules change, then now this monkey should not choose yellow. So this monkey is, want to really pay attention to what's happening to the other monkey before his own turn. And this is just quantification of how much they lick at the juice tube. When partners make correct choice, after making a choice, the monkeys start licking a lot because in this test, both monkeys always get juice when they're right. Both monkeys never get juice when they're wrong. So this licking pattern, it's like a conditioning response to start licking. But when other monkey makes an erroneous choice, then the licking drops out. So they understand what's going on. So they recorded from medial frontal cortex, a little bit more posterior than others previous study, but they have the sulcus of the ACC part and also this part, which is kind of pre-supplemental motor area, basically. And what you see is that you see neurons like this, a lot of them, partner action neuron. This neuron was only exclusively activated when partners make a choice, but basically nothing when self makes a choice. And when you average all these kind of neurons, and you see this nice pattern that partner action, self-action. And you see that, partner type, are, plenty there. Almost, slightly comparable number or percentage as a self-type. In the same area, they also found neurons that encode other monkeys' error. In this case, this is partner's erroneous choice. This is aligned to the button press when partner monkey made an error. This neuron fires really strongly. Whereas the same neuron, but when partner made a correct choice, there's not much activity at all. So if you average across all these kind of cells, and you see this partner type error specific activity in both of these regions. I mean, there are neurons, signal partner's correct choice too, like what you saw before. But these, 17% of cells were completely specific to, strongly specific to other monkeys' error. Finally, the competition is a very important component of social decision making. So in my, I don't know, maybe some of you missed this kind of game, but I grew up when the video games kind of started, and Missile Commando was a kind of very popular game. This is how it works. It's a very simple Atari game. And you're trying to, shoot these things without shooting your own supply plane. So it's a very simple game. People loved it. It's an Atari game. I bet you can buy it now too, even. Anyway, this group from Japan basically have monkeys play Missile Commando. And I like this study a lot because they did such a good control to rule out various things. So basically, the one condition, two monkeys are playing this Missile Commando. This monkey is trying to shoot this guy. This monkey is trying to shoot that one. In the control, you play with a computer. There's also just a steady target, just a performance condition. And target location can be one of these. And then monkeys, shoot, and whoever hits the other target first are the one get rewarded. If the other monkey gets your target first, you don't get rewarded. Okay. They recorded from lateral prefrontal cortex, and they found in majority 3 types of neurons. This is shown as this column, this row is the 1, meaning that actor monkey 1, the recorded monkey 1, and this is when the recorded monkey lost. And this neuron is they call the win competitive plus because they care about winning, this one, but not here. But they care about competition. When there's no competition with a real monkey, the same neuron fired a lot less. So this neuron was specific to winning against a real monkey partner. This was lose competitive neuron, meaning that this neuron signaled more about when you lost to the other monkey, but not when you lost to the computer, for example. This is win-lose competitive neuron. They don't care whether you won or you lost, as long as it's in a competitive condition and some outcome happened, these neurons fire a lot more than non-competitive condition. The presence of real partner really mattered here because they compare monkey-monkey and monkey-computer competition. You see cells that are specific to, specific to competing and winning, specific to competing and losing. So another control they did was very interesting. So you also had a monkey in the same room, but this monkey is not playing anymore. So they're kind of controlling the just general arousal factor, but monkeys are there but not playing, and you see very similar activity here. So you have these 4 different kind of settings to look at this activity, and they generally find the competition drives a lot of activity, and also partner matters. So finally, I want to talk about amygdala. We have like 5 minutes left. Amygdala is a key structure for motivation and emotion, facial expression, internal personal space, also implicated in autism, for example. Just going back to the patient SM idea, you have a interesting thing is that you have this deficit in looking at the eyes. But now, when you ask patient SM to look at the eyes and judge emotion, then she becomes completely controlled. So which suggests that amygdala it's not that amygdala is needed to judge, in this case, fear, but it's that, without amygdala, you just don't know how to allocate your social attention correctly to judge the fear. Because if you tell her to look at the eyes, you do it really well. Another reason to talk about amygdala is that if you do international oxytocin study and put people in the scanner, amygdala is the most common area to change activation, usually go down. So we recorded amygdala in this neuron, in this task. And this is just the same preference again from this task. Prefers other, prosocial preference, prefers self, antisocial preference. In this case, we had this reward value queue to change the amount of juice. And you can see that choice reaction time reflects that. So monkeys are faster when the higher juices come. Just a proof of concept. In the amygdala neurons, what we see is that when monkeys make a choice, you have a very similar activity across different reward outcomes. But what's interesting is that there's some interesting same scaling of juice value. So if you plot the correlation, if a neuron is tuned positive to your own reward, it's also tuned positive to the other reward, and also for the both reward. This was not the case when you compare to the neither reward. So in this area, there is doing some kind of value co-tuning. If something's valuable to me, it's also valuable to you in that neuron's perspective. If something's not valuable to me, also not valuable to you in that neuron's perspective. The very last thing I want to show you is that we talked about amygdala and AC gyrus. We asked what are they doing in communication? Are they talking to each other? So we recorded from both areas simultaneously. We use this axial array, multiple channel array, so that we can record a bunch of neurons together. And one of the reasons to do that is now we can also look at how the 2 areas are coupled to each other, talking to each other. And then you can look at the field potential oscillations here. That's the sample trace, and now you can, decompose this to understand how the, oscillatory activity across the two areas are synchronized or not synchronized. So when you do this kind of thing, people call this field-field coherence. When you do this kind of experiment, you see something interesting. What we see is that this is a coherence spectrogram of when monkeys choose prosocial preference, express prosocial preference, compared to when monkeys express antisocial preference. What's showing here is that red means amygdala and the ACS gyrus are much more phase-locked to one another, compared to blue means they are not phase-locked to one another. It's aligned to the time of choice, and what you see is that right before making prosocial choice compared to antisocial choice, gamma band frequency domain have a higher coupling synchrony compared to beta band. Beta band synchrony actually decreases. So when you quantify this, now I'm plotting frequency here and the coherence value over here, you see that around this time, you have suppression in beta band synchrony and enhancement in gamma band synchrony. This is just a subtraction of this prosocial choice and the antisocial choice to get a contrast. And it turns out, this suggests that, well, gamma is going up and beta is going down. Are there some interesting relationship here? So we looked at gamma to beta ratio for choosing self, both, other, neither. And it turns out when monkeys are choosing other to give to the other monkey, the gamma to beta ratio was indeed the highest. So we think this is a possible signature prosocial decision in this context. And we don't have time to show you today, but if you, infuse oxytocin to amygdala, this gets stronger, for example. There are a lot of interesting signature around gamma to beta ratio. So to conclude, we talked about dynamic and contingent nature of social attention, and I think that's a very important prerequisite to thinking about other minds. And also context-dependent prosocial and antisocial decision, that's a way to quantify how you think about other people's mind. Without this kind of behavioral output, it's really hard to quantify, right? And finally, there's some really similar neuromechanisms happening between human and monkeys. I think then the question is, what's the difference between monkeys and humans? Monkeys are definitely not humans, and their social behavior is definitely not sophisticated. I think what might boil down to is that we are much more complex in terms of how abstractness we can represent this kind of information. So basic information might be all there in the monkey brain, but a monkey might not have the similar kind of way to pull those to create more abstract information in the higher dimensional space for more complex social behavior. I think that might be the difference, rather than say humans have this process, monkeys don't have it. It's not. I don't think it's clear qualitative difference. I think it's more of a computational limitation in monkeys to make those very abstract social representation. Okay, thank you.
The findings are very interesting, and they certainly fit with the theme of the summer school. But I have to ask you, what are the prospects of being able to do this get this kind of information non-invasively?
That's a very important question. Actually, we always look for non-invasive way to do this kind of research. And so what's invasive about this kind of work is that, first, craniotomy, right? You have to open up the skull to get in there. In order not to do that, you have to use non-invasive technique like functional neuroimaging. The problem of that is the spatial resolution and the temporal resolution is very, very slow. You cannot get insight into computations. Neurons fire in a millisecond resolution. fMRI is like you have 2 minutes lag time. Also, spatial resolution 1 millimeter. In a 1 millimeter voxel, you have 100,000 neurons. So that's that's the scientific limitation there. And the only time, so people are starting to now collaborate with the neurosurgeons to study people going through neurosurgery to record from their neurons. And actually, a lot of patients are very happy to do that. It's like, neurosurgery, you're awake, you're bored, and a lot of times they want to play a game, and when you're identifying seizure locations, you have to record data anyway. So there are those kind of studies out there, but of course you cannot really have a good control in the environment and the repetition and such. And also the rodent model has a lot of strong advantages, although very invasive, more so. But rodent model, the one thing is that behaviorally they're often different. So there's and the prefrontal cortex is often different. But they can do a lot more sophisticated circuit kind of thing. And also the another kind of non-invasive procedure people can think is, people are starting to use eye tracking without head immobilization. But that's still kind of, you can do behavioral tasks with that fine. But the question is when you're recording from the brain, any movements can kind of get the cell go away, basically. So there's a lot of limitations in those techniques. And people are also putting a lot of efforts towards, in silico experiments. For example, in the early visual areas, we know from long time of a lot of work, we know a lot of important neural encoding in the vision domain. So some of those we can start working on some computer, but a lot of this high-level cognitive behavior, there's really not much knowledge out there. So I think, what people try is, to be as less invasive as possible, and when it's invasive, only really be mindful about when you're collecting data. What are you going to do with a monkey after collecting data? So there's a lot of concerns like that. And I think these are real issues. So people are constantly trying to develop much more non-invasive way of doing this kind of experiments. So in the future, if the imaging technique becomes much higher resolution, then they might be able to overcome this kind of problem.
Hi, thank you for the talk. I just want to know if I understood correctly the experiment with the Atari game. So the monkeys really are engaging in the competition. It's only for the game. There wasn't anything else.
That's right.
Oh, wow.
Yeah, so
Okay.
So interesting about monkeys, they are in the wild, they're very competitive. These monkeys are, rhesus monkey especially is the highest, most despotic macaque species. There are 19 species. The rhesus is at the top. They're very despotic, but they're competitive over resources. And in the lab, when you train them, if you make the task competitive, they train so much faster because it's just natural to them. But when you train to be prosocial, it's very hard. So I think, competition is very natural, and, you do it, if you lose, you don't get reward. So you're very much motivated to, get reward.
Okay, so for the game, there was a reward at the end. It wasn't only for the ludic
right.
aspect
So
of it?
they play many trials of this game.
Okay.
Every trial, the winner gets juice.
Oh, okay.
The loser don't get the juice.
Okay.
Yeah, that's what's motivating.
Okay,
So in analogy, in rodents, they get pellet, right? In birds, they get some pellet kind of thing. But monkeys, the fluid is a good motivator.
Okay, thanks.
Yeah.
Hi. I just saw one of your papers where you talked about implanting head posts. And I know it's still a standard practice, but there are alternatives. And so I'm wondering if you had thought about either there's a helmet now, and then there's also thermoplastic mask.
That's right.
And then you can also, I don't know how to say that in English, but make a cast of every individual skull, and then you can use that. Because just as an experience as a veterinarian, I saw many infected head implants.
Sure.
And I I just think this is unnecessary. And
I actually know those. Actually, was involved in developing one of those when I was there at Duke, for example. The one was developed at Duke, the thermoplastic mask.
Yeah.
We decided not to try it because you cannot do social experiments with it. Because social experiments, you cannot hide your face, because that mask there's eye and mouth hole, everything is covered.
Okay.
So that was out of the question for me. However, the aquaplast molding, we tried and we use it actually. We can do behavior with it, but we have a hard time doing recording with it because behavior, you can slightly move like this and still get the data with eye movement tracking. But for recording from neurons, even tiny movement can make the data not analyzable. So we do have that as an option for behavioral training in the lab. We spent quite a long time developing it, and it's it takes time, but it's definitely worth it. Some monkeys, what we tried was we had a really old aged monkey for aging study. So old aged monkey, you cannot put head post, right? Because their bones weak. So we tried that mask kind of thing for behavior. So we have that as in the lab as a backup use, but for when you get to the recording level, it's just not practical anymore.
And did you try the helmets? I mean, I can send you the papers on that if you're interested.
So we didn't try the helmet, no. Yeah.
Because maybe you find some other option, because it's just very invasive. I don't know who came up with this idea to implant something in an animal skull just to fixate them in a monkey chair. I think it's just out of imagination.
I mean, people are trying many things, but we didn't try the helmet. But the aquaplast is behaviorally good. Maybe helmet will be even good for recording, right?
Yeah, I will send you the papers.
Yeah, thanks.
More questions?
Anyway, let me ask, there's a self-other sort of reciprocity over here. Can you relate that to thinking that you haven't been present, but thinking that we've had about perception and production systems, and also, of course, the famous mirror neuron story?
Yeah, that's a great question. I didn't put mirror neuron here because, actually, I think I should mention it now. So mirror neuron is very famous. It's there in the brain, but it's mostly in the motor areas, actually. It's not found in this kind of high-level prefrontal cortex area. Mirror neurons hasn't been yet to be demonstrated as really socially relevant. Even, mirror neuron became popular very fast, almost too fast. I think same thing I can say the same thing for oxytocin. It became popular all of a sudden too fast. But mirror neurons, there's a group by David Heger in NYU that did a really excellent study, in my opinion, to really say people were hypothesizing, mirror neuron might be what's going on in autism, something weird with mirror neuron. So that group did a really beautiful study, comprehensive study to demonstrate the mirror neuron system in autistic kids were completely normal. So, how mirror neuron links to actual social behavior is still unknown. But it's clearly there. We don't know. And the one alternative hypothesis is that maybe mirror neuron is not really about social behavior, but it could be about more motor simulation. That's more non-social explanation of it. I have no strong views of this, but in my opinion, I don't know whether we actually know mirror neuron is either involved in directly social thing, or are you rehearsing a lot of these motor dimension movements? So that's that. And in terms of the this kind of task, no one has ever recorded in the mirror neurons in monkeys in this kind of task. When people do in humans, because humans also have mirror neuron system, and when people do this kind of social decision-making task in the scanner, Mirror neuron system in the premotor area, for example, are not always involved. What's more involved is the valuation system in the prefrontal cortex and the temporal-parietal junction kind of system that are considered to be important for theory of mind. So, even in the imaging literature, maybe it's because the task, making nice gestures to other people kind of task, maybe that's not what's engaging mirror neuron, and mirror neuron might be still involved in social cognition, but that's not engaging it. That's a possibility. But so far, the stronger hypothesis, the valuation system, seems to be more robust in social behavior, coupled with more of a, allocentric temporal parietal junction, which processes like biological motion, certainty of an event, for example. So I think mirror neuron evidence is very interesting because although it's a small in number, those small cells are very, very robustly clear what they do. We just don't know how that links to other behavior. And also, lesioning the mirror neuron system is tricky to see what happens because it's not it affects other, because a given area like a ventral premotor cortex does a lot of things with other behavior as well. So if you just inactivate or lesion those areas, it's going to be hard to tease apart whether it's mirror neuron because of mirror neuron or not. So, one beautiful study would be, using in rodents, like using optogenetics kind of thing to specifically target certain cell types. But as far as I know, rodents don't have mirror neurons. Or maybe maybe someone thinking about how to test mirror neurons in rodents. But if rodents have mirror neurons, maybe they can selectively manipulate those and see what happens. But at this level, at this time, I think, there's no strong evidence toward or a little bit more evidence against mirror neuron probably for social behavior.
Are you aware of any studies, or if there are none, do you have any thoughts about the feasibility of studies where things like oxytocin, things that mediate prosocial and self-regarding behavior, could be administered chronically to all members of the group to see if the group arrives at a different stable pattern of social relations? Yeah, so those, and only study I know about chronic oxytocin is in rodents. So there's a famous study that shows that if you give oxytocin for too long, actually, I think it's rats became more aggressive to the, partners. So that was often cited as one of the papers to potentially telling us to, telling, clinical trial and those kind of fields to slow down a little bit because we still don't know what's the effect of long-term chronic effect of oxytocin. So in monkeys, it's really hard to study in long-term oxytocin because some of these monkeys are involved in many different behavioral tasks. It's hard to, it's almost like cost prohibitive to have monkeys just do one thing forever. And however, in the wild, there's a correlative examples that there are some examples of changes in oxytocin level as a function of aging, but also there's also lack of evidence of oxytocin in controlling where in the social network you are, for example, because in the field you can, if you have 50 monkeys, you can actually make a social, using a graph theory to make a social network and see how different genetic background and stuff like that is modulating where you are, center or the periphery. We know that serotonin polymorphism has something to do with how central you are. If you have a short artery, you're more likely to be out in the kind of less popular part. When people looked at for oxytocin genotype, they didn't find anything. So, the effect on how oxytocin affects large group is unknown because it's so hard to do causally. In an ideal situation, maybe you have, 50 monkeys in a colony, and I'm gonna randomly select a monkey and increase their oxytocin, give them a lot of oxytocin for a long time, and see how the rank changes. But that kind of experiment is just not practically easy, and also there's no good place to do it.
On the other hand, in the laboratory situation, you know, in the laboratory, it's not like you have 20 animals all living together interacting. They're, you know, in pairs and group housed in a smaller groups. So much harder to do that experiments. That's a good question, though.
Thanks.
Yeah. Thank you.